This is Tasha Santiago Rodriguez. She's a scientist here at the ATCC Center. Dr. Santiago Rodriguez got her PhD at the University of Puerto Rico in public health water microbiology. Her postdoc at UC San Diego was on the characterization of metatranscriptome of MRSA, bacteriophages in the human bloodstream, microbiome and virome in association with urinary tract infections, and short and long effects of antibiotics to the human virome. She was a recipient of a fellowship from the Howard Hughes Medical Institute to work in the characterization of ancient human microbiomes, viromes, and resistomes. Thank
you. Hi, everyone. It's really a pleasure for me to be here and talk to you about the gut microbiome and resistome of ancient cultures. And I'm going to be talking particularly about the gut microbiome and resistome of the Incas and ancient European nobility. As all of you know, the gut microbiome and gut microbiome studies have been characterized a lot during the last couple of years, mainly because we have the technology and the bioinformatic tools more accessible. We know that the gut microbiome has been characterized in association with health and several different conditions and diseases, such as autism, inflammatory bowel disease, diabetes, cancer, just to mention a few. But what about ancient gut microbiomes? So I have had the opportunity to work with both, let's say, modern gut microbiomes and ancient gut microbiomes, and there is information you can get when you have access to ancient gut microbiomes that you can really get when you have access to, let's say, modern gut microbiomes. For example, you can study more directly the ancestral or the original state of the human gut microbiome. And by this, I mean that you can see how was the microbiome before, let's say, a westernized lifestyle or a westernized diet, or even before antibiotic therapy. You can study in real time the evolution of both commensal and pathogenic microorganisms. You can study the evolution of functional profiles and metabolic profiles associated with the human gut microbiome. You can study the evolution of antibiotic resistance, or the resistome, which is the term we use to refer to all of the antibiotic-resistant genes in a particular sample type. There are some disadvantages, though, when you work with ancient gut microbiomes. Even though there is people that feel very excited and very intrigued about this type of studies, there's also a lot of skepticism out there, mainly because of contamination issues. The DNA tends to be highly degraded, and this tends to limit some of the analysis you can do. But this is actually one of the characteristics of ancient microbial DNA. It has to be highly degraded. If you get DNA that's intact, then you know that you probably have some contamination issues. You usually get small amounts of DNA to start with. When you sequence, you can get very short reads and contigs, and this can sometimes result in having low similarity to sequences present in existing databases. Ideally, you would process and handle your ancient DNA sample in an ancient DNA laboratory. I know of some scientists that don't really have access to an ancient DNA laboratory, so what they would do would be to have an area within the lab that would be exclusive for ancient DNA analysis, and everything has to be dedicated for ancient DNA analysis. The pipettes, the reagents, the lab coats, everything has to be dedicated only for ancient DNA. We would then send the samples to get sequenced in a sequencing facility, and we would do, for example, 16S high-throughput sequencing on shotgun metagenomics using Illumina MiSeq. And we would do some of the classical profiling, such as taxonomic profiling, metabolic profiling, resistome profiling. For the taxonomic profiling, we would use, for example, SHIME. We would use CLAMS, CLC Genomics Workbench, just to mention a few. For the metabolic profiling, we would use, for example, MGRAS, Diagrid, CASI, PyCross. And for the resistome profiles, we would do a blast analysis against a database that we know is updated and curated for antibiotic-resistant genes, such like CARDS and ResFAMs, just to mention a few. But I would say one of the most important things is how do you interpret that data and what story you can make out of that data. Some of our most recent research has been highlighted in the media. For example, there was a study where we characterized the gut microbiome of two cultures that used to live in Puerto Rico about 1,500 years ago. And we were able to show by 16S analysis that these two different cultures have very different gut microbiomes. And we think that these differences are due to differences in culture and diets. Probably a year ago, our study about the gut microbiome and resistome of a pre-Columbian Andean mummy also got highlighted in the media. And probably a couple of months ago at the ESM meeting, our study about the microbiome and resistome of the Incas and ancient Italian nobility mummies also got highlighted as well. And this is the data that I'm going to be talking to you today. I guess when someone talks about the process of mummification, we tend to think about Egyptian mummies and the process of artificial mummification, but there's also the process of natural mummification that results from the exposure to dry and cold environments. This will result in the water content to decrease from about 75% to about 25% or so. Any enzymatic action associated with the process of putrefaction will be minimized or halted. The process of natural mummification was practiced in ancient civilizations. For example, in South America, the Incas, they would, for example, place a body inside a basket, and they would place that basket somewhere outside in the environment where the conditions were good enough for the process to happen. And also in some regions in Europe, even though it was different, they would place the body inside a chamber that had the conditions ideal for the process to happen. Currently, we have access to the DNA of 3 Inca mummies. One of the mummies, Mummy FI9, she actually wasn't from the Inca period. She was more from the pre-Inca period. She lived in the 10 to 11 centuries. The mummy was very well preserved, with the exception of the face that was skeletonized. The rest of the body was very well preserved. She was about 18 to 23 years old when she died, and our pathologists think that she died from Chagas disease, or at least suffered from it. Chagas disease is endemic from Central and South America, and it's caused by Trypanosoma cruzi. Mummy FI3, he was from the Inca period. He lived in the 14th to 15th centuries. The mummy was also very well preserved. He was about 25 to 30 years old when he died, and it's possible that he suffered from leishmaniasis, which is caused by Leishmania donovani. And Mummy FI12 was a female from the 14th to 15th centuries. The mummy was also very well preserved. She was about 20 to 25 years old when she died, and it's possible that she suffered from bronchopneumonia. Just to give you an idea of where these mummies come from, they come from Cusco, which is the ancient capital of Peru, located in South America. And the Incas had a socioeconomical division of classes where the commoners or the farmers were at the bottom of the pyramid, followed by the nobility, and at the top of the pyramid we have the emperor. But despite this division of classes, the Incas had mostly a vegetarian-based diet. They ate a lot of crops, a lot of potatoes, a lot of corn, a lot of peppers, quinoa, just to mention a few items, and they rarely consume meat. We also have access to the DNA of 5 European nobility mummies. These mummies were found in Naples, in Italy, Europe, and we know who these people were. We know that we have dukes, we have a king, we have a prince. They lived in the 15th to 16th centuries. Most of them were males. We know we have a mummy that died when he was about 12. Another one died when he was about 70, and we know that they suffer from different conditions like bronchopneumonia, different types of cancer, cirrhosis, just to mention a few things. And also I have to highlight that the Italian nobility, unlike the Incas, had mostly a meat-based diet. They ate a lot of meat, and rarely they consumed fruits, and rarely they consumed vegetables. So given that we had access to these very different cultures, the Incas and the Italian nobility, with differences in gender, with differences in age, pathologies, lifestyles, dietary habits, and socioeconomic status, we hypothesized that the microbiome of these mummies, the functional adaptations or the metabolic profiles, and the resistomes may be associated with culture and even maybe in some cases subject-specific. Remember that I mentioned very, very briefly that you always need to make sure that what you have is what it is, that you have authentic ancient microbial DNA. And what you can do is, for example, run a gel and see how degraded your DNA is. If you have intact DNA, then you probably have some contamination issues. I don't have a picture of that, but this is a picture of a PCR amplification of a human papillomavirus that was detected in one of the mummies. And as you can see, at least in this lane, there is a band there, even though it's very, very light. This is consistent with starting with very small amounts of DNA, which is one of the characteristics of authentic ancient microbial DNA. Also, you can do some bioinformatic analysis. This one is called Source Tracker, where you would compare your 16S sequences with your selected microbiomes. In this case, we wanted to compare these 16S sequences with modern gut, oral, skin, and soil microbiomes. We were more concerned with contamination with soil microbiomes because to some extent, these mummies were exposed to the environment. But as you can see here in these pie charts, really there aren't any sequences being classified as soil microbiomes. In most of the cases, as you can see here in these pie charts, with the exception of mummy FI3 that had about 85% of the sequences matching modern gut microbiomes, in most of the mummies, more than 95% of the sequences didn't match any of the microbiomes we included in this particular analysis. But this is actually consistent with a previous study that was published in PLOS ONE by Tito et al. In 2012, that they characterized the gut microbiome of Otzi, this very famous mummy, and they also found that the gut microbiome of Otzi didn't really resemble any of the modern microbiomes they included. So then we proceeded to do some taxonomic profiling, and based on the 16S data, there is a separation of the data, as shown here in this Procrustes analysis, based on culture. Here in this purple color, you see all the Inca mummies, and here in red, you see all the Italian nobility mummies. Then we proceeded to do a heat map based on the relative abundances of selected bacterial families. And as you can see also, there is a separation based on culture. Here you see in black all the Inca mummies, and here in white, you see all the Italian nobility mummies. It's also very interesting to see that, at least in the Inca females, more than 95% of the sequences were classified as Clostridiales. In the Inca male, more than 95% of the sequences were classified as Bacillales. And in the Italian nobility mummies, it's very interesting, at least for us, to see that they had a higher bacterial diversity. We saw not only the Clostridiales or Bacillales, we also saw, for example, Sphingomonadalis, just to mention a few of them. We also performed some taxonomic profiling based on shotgun metagenomic data, and as you can see, the profiles don't really resemble the 16S profiles, but this is not an issue from ancient DNA analysis. This is something that has been highlighted before by other researchers. But there is some separation of the data based on culture. Here you see in black all the Inca mummies, and here in white you see the Italian nobility mummies. But the take-home message is that it seems like the process of natural mummification preserves bacterial DNA from different families, not only gram-positives as we thought originally, but also from gram-negatives. And this will hopefully open the opportunity to understand some of the mechanisms involved in microbial DNA preservation. And also, I didn't show this data today, but the process of natural mummification also preserves viral DNA like bacteriophages, human papillomavirus nucleic acids, and the nucleic acids of pathogenic eukaryotes, such as Trypanosoma cruzi and Leishmania donovani. We also were very interested in the metabolic profiles of these mummies mainly because a lot of studies associated with the gut microbiomes, at least at the beginning, they tend to concentrate only on who is present in a particular sample, which bacterial communities particularly. But it's also very important to understand what are some of the things these microbial communities are doing in some of these samples. In our case, we wanted to see which of these metabolic genes are being preserved during the process of natural mummification. So we noticed genes associated with different types of metabolism, like sulfur metabolism, RNA metabolism, protein, carbohydrate, amino acid metabolism, just to mention a few categories in both the Incas, here shown in blue, and the Italian nobility mummies, here shown in red. We didn't find any significant differences. We think that it's due because in the Italian nobility mummies group, we had a mummy that was about 12 when he died, another one was 70, so we think that the age difference might have been one of the factors that influenced these results. We also were interested in carbohydrate active enzymes, or Cas-enzyme profiles, mainly because carbohydrates are an essential component of any culture's diet, so it's very important to understand some of these Cas-enzyme profiles because they are involved in the degradation of both simple and complex carbohydrates. I'm not showing you all of the data because it gets a little bit complicated. But for example, here in panel A, I'm showing you the average number of all of the Cas-enzyme genes we found in the Inca mummies, here shown in black, the Italian nobility mummies, here shown in white, and also we wanted to compare the data with modern groups like the modern Amazonians. We showed this group because they resemble to some extent the Incas, and also here in red, modern Europeans, which are actually modern Italians. And as you can see, the Italian nobility mummies had a higher proportion of Cas-enzyme genes. Here in panel B, I'm showing you Cas-enzymes with auxiliary activities. These are involved in plant cell wall degradation, and as you can see, there was a higher proportion of these in the Inca mummies compared to the other groups we included. In panel C, I'm showing you glycoside hydrolases. These enzymes are involved in the degradation of more complex sugars, and we didn't find any significant differences between the groups. We also were very, very interested in some of the putative resistome profiles. Remember that I mentioned resistome is the term we use to refer to all of the antibiotic-resistant genes in a particular sample. And I have to admit that at first we were very surprised. We told new scientists at the last ASM meeting in Boston, but then it's at least funny for me to read what Raul Cano said, and for those that know him, know that this is something he would say. He said, "It's ridiculous to think that the evolution of antibiotic resistance began when penicillin was discovered about, 80-something years ago. It's been going on for 2 billion years. I don't know if it's been going on for 2 billion years, but I know at least that the data is showing that it's been going on for at least 30,000 years, like this paper that was published probably 5 years ago that found putative beta-lactamases in permafrost dating to 30,000 years. The Incas, here shown in blue, had a higher proportion of putative antibiotic-resistant genes compared to the Italian nobility mummies, here shown in this orange. Both of them had a lower proportion of putative antibiotic-resistant genes compared to the modern groups like the modern Amazonians, here shown in gray, and the modern Italians, here shown in this yellow color. We are still digesting this information, and we have been thinking a lot about it, especially from a microbial ecology point of view. And we have come to hypothesize that a greater exposure to the environment may be associated with a greater acquisition of putative antibiotic-resistant genes. So remember that the Incas, to some extent, were more exposed to the environment. Most of them were farmers, even though we do not know if our mummies were farmers or not. We know that at least they consume crops that to some extent were contaminated with soil microorganisms. And this is something that a lot of people are thinking about. I really don't know how to really prove this information other than doing microbiome analysis, but this is something that a lot of people are thinking about. If we break down this data into even further categories, we notice that there are some genes that resemble some modern putative beta-lactamases, here shown in blue. some chloramphenicol-resistant genes, here shown in yellow, some multi-drug transporters, here shown in red, some tetracycline-resistant genes, here shown in this brown, and some vancomycin-resistant genes, here shown in this navy blue. We are still, this is very preliminary data, and we are still digesting this data a lot, and we are making some of the analysis to see if there are any significant differences between all of the groups we included. But for now, the take-home message is that we think that putative antibiotic-resistant genes may not only be associated with the use of antibiotics. So I want to conclude by restating that ancient DNA has to be suitable for microbiome analysis, and this has a lot to do with how the sample is collected, how the sample is processed. You need to authenticate your DNA. You need to make sure that what you have is what it is, that it's authentic ancient microbial DNA, and for this you can do an agarose gel verification. You can do some bioinformatic analysis like source tracker, and always include some type of control during the DNA extraction, during the PCR amplification, during library prep, if possible. And talking about controls, there is a survey on your tables, if you can fill it up. It's just for us, about controls. But when you do have access to this type of samples, they can provide information about the ancestral or the original state of the human gut microbiome and how it has evolved, at least during part of human history, and how it might be associated with different dietary habits, differences in lifestyles. Here we saw the Incas versus the Italian nobility, but if you have access to other ancient cultures, it's very possible that you can do very similar analysis, and also in association with different pathologies. For example, I haven't really seen a paper that has characterized the gut microbiome in association with these neglected tropical diseases like Chagas disease or leishmaniasis. And in terms of the resistance profiles, even though we are still digesting this data, it will open the opportunity to elucidate the evolution of antibiotic resistance and possibly predict the evolution of antibiotic resistance. So I want to finish by acknowledging the team of scientists involved in getting these microbiomes characterized. Dr. Gino Fornaciari here at the top from the University of Pisa in Italy. He is our pathologist, the one that collects the sample and has the hands-on experience with the mummies. Dr. Stefania Luciani and Isolina Marotta from the University of Camerino in Italy. They are the ones that make sure that this DNA is highly degraded. My ex-PhD advisor from the University of Puerto Rico, he is the one that tries to make sense out of this data from a microbial ecology and possibly public health point of view. And also, of course, Dr. Raul Cano for trying to merge ancient DNA, mummies, and gut microbiomes together. He was my postdoc advisor for a year, and now he's my boss. Also, I want to acknowledge Cal Poly, where I did a lot of the data analysis, the Life Sciences Research Foundation, the Howard Hughes Medical Institute for funding, and also I want to acknowledge all the people from ILSE and the ATCC Center for Translational Microbiology. Any questions?
I might have missed it, but how do you know that you don't just have bacterial overgrowth after death, or what during the mummification process stops?
Yeah, so that's a difficult question. We think that a lot of these profiles resemble initial stages of decomposition. I've seen papers that have characterized the gut microbiome during different stages of decomposition, and they kind of resemble earlier stages. But we're not really sure how to really show that.
I guess a related question, maybe naively, is when somebody's embalmed currently, do they flush out their intestines, or can you do this in
I don't know.
more recent people than ancient?
I don't know. That's
So
a good.
you
I
have no
don't
way
know.
to look at recent deaths then?
Good question.
I don't know.
Thank you.
So how much of a problem have you had with your reagents? I've heard horror stories of people's enzymes or whatever reagents for all of this analysis being contaminated and coming up with sequences that are totally unexpected. Have you had that as a problem?
We have, but then we just then make sure to just buy new reagents. But what we would do, for example, is sequence, for example, a control, and if something comes out, you can filter out those sequences. Mm-hmm.